Good morning, everyone out there in YouTube land. So the micro class does get weird-ass random comments. And hopefully by the time we're done, we start getting into a little bit of cancer biology. This class will get some weird unsolicited comments as well. I'm surprised we're not getting anything about diets and metabolism. As I say, I've been looking up dinitrophenol to see if you can actually still get it online. And I think you can. It's quite dangerous. We shouldn't take it. But, all right, so the exam is still in the works, but it's going to be pretty much the same format. I'm not sure how many multiple choice questions we're going to do. So at least on video, we've been covering through all of our metabolism stuff. We went over the worksheet in the lab. And I don't know, we probably should get into the new topic stuff, but I always feel bad about that. So we're going to just do a little bit more backing and fill and kind of start from where we were at the very beginning. Because first off, we were looking at the control of gene expression. Is where we began this part of our journey. And we're actually going to get back into some molecular genetic things when we start getting into secretory pathways. But, you know, some basic stuff here. Control of gene expression, we have two different kinds of models that we can think about based on the different kinds of systems. The prokaryotic basic model, because RNA polymerase does have intrinsic binding affinity to DNA, and the sigma factor directs binding affinity to specific promoters, we have this idea of well, the operon, which is a set of genes, and then the operator, which, unfortunately, sort of names and things, it is a DNA binding spot for some kind of a repressor or, yeah, let's go ahead and keep it simple like that. So we have the repressor binding spot, and that usually is going to be somewhere, you know, around the minus 35, minus 10 site, and often blocking +1, because the RNA polymerase with the sigma factor has intrinsic binding affinity. If you've got some kind of great big protein sitting here, it can't bind. So that's one of the ways we can think about the gene expression, is we have a repressor, and then it's going to bind a small molecule. That could be an amino acid. It could be a molecule. Sure, molecule. It could be a sugar, whatever. And depending on how we want to play this, the protein in the presence of the sugar would fall off. Maybe if we're trying to do some sort of biosynthetic thing, the protein in the presence of the biosynthetic product will bind, and when that biosynthetic product comes off, it falls off. But it's the basic theme, is that we're blocking the progression of the RNA polymerase. So we can just sort of, as a generic way, we can block progression of RNA polymerase. Now, another variation on this theme, and we're kind of like thinking about the lac operon as well, but another variation of this theme is we can have some way that the binding affinity for the promoter isn't very good, i.e. we have a bad minus 35 site. So we can alter the binding affinity to the promoter with some kind of factor, some kind of control system. And what this does is actually sets us up for the complete story of the lac operon, which, let's go ahead and throw that out there. The lac operon is a really good model system, but a lot of systems work on a variation of this theme, in which we've got a constitutive produced lac repressor. So this, actually in the parlance, is known as the lac-I gene. This is the repressor, which binds to the operator sequence. Lac-I, this guy is constitutive. And we're going to go ahead and put its binding site right here, kind of straddling +1. All right, so this binds. And now we've got a -35 site over here. RNA polymerase is going to try to bind at its -10 site here, and it can't proceed through unless that repressor is gone. The other little tweak to the lac operon is this. The -35 site isn't real good, and so we need to have a second kind of protein present in order to actually fire this thing. And so that requires the introduction of the catabolite activator protein, or the CAP protein, whatever. The catabolite activator protein. This is able to bind at its site, kind of right here, and it now is able to recruit RNA polymerase and basically force it to bind at a less than ideal -35 site. Now, the CAP protein, this is only turned on, induced, or active. How about this? It is active as a DNA binding protein in the presence of cyclic AMP, which is basically the alarm signal of saying we don't have anything to eat. So cyclic AMP is coming on saying there is no sugar metabolites for us to chew on. That in turn activates the CAP protein. CAP is able to bind here. If we have lactose, the inducer, it can bind here. And that causes lac-I to fall off. And so only under these two conditions, low amounts of sugar and the presence of lactose, actually I guess low amounts of glucose, and the presence of lactose, will this thing actually fire at its maximum level. So that's why if we've got this sort of diagram of glucose and lactose, if we've got lactose in the media and glucose in the media, the lac operon is still off because we're going to be consuming glucose. If we've got no glucose but lactose is present, so here it's off, now it's on in a really big way. If there is no glucose and no lactose, well, things are just sad, and the system is still off because, as we said, the production of that protein complex, all those different enzymes, is metabolically expensive. There's no reason to have them on because they don't have anything to be chewing on, and so the system is still off. If you've got glucose present and no lactose, well, yeah, you're just eating glucose because that's what everybody likes to eat. So that explains the system fairly well. And now we can actually adjust this for different kinds of systems. So in the case of like tryptophan biosynthesis, and actually the trip operon's got some very clever little tricks to it, which we talk about in molecular genetics, we won't talk about here. The trip operon is working in a similar kind of fashion, but now instead of having the sugar molecule being the binding factor that causes this thing to come off, tryptophan, as long as it's present, that means you don't need to be biosynthesizing tryptophan. Tryptophan being present is making the repressor function and stay on its operator site. And it's only when tryptophan levels drop below a critical spot that this thing is able to now, tryptophan diffuses off, the repressor falls off, and you can get read-through. And tryptophan biosynthesis doesn't require the same kind of juggling, because it's not like if you've got a better sugar substrate, keep this thing off. Tryptophan gets low, you got to turn on your tryptophan biosynthesis. There are a couple of the tricks that tryptophan operons and amino acid biosynthesis operons use just as a fine tune, but this works pretty well. So this, in a generic fashion, is the scheme by which bacteria are controlling gene expression. And it's really based on the fact that RNA polymerase by itself, the alpha-2 beta-beta prime sigma factor complex, is going to bind to the promoter. So really all you have to do is block this. Now, in the prokaryote, or the eukaryotic system, we have a different kind of game, because RNA polymerase in the eukaryotic world doesn't really bind particularly well to the DNA itself. What we have to have for that is the basal transcription apparatus, as is known. And this is binding to its various sites and establishing a little nest for the RNA polymerase. And we're specifically talking about Pol2 here. And so Pol2 can bind with all these other ancillary proteins here, grabbing it, hugging it, and it begins to transcribe. So the primary thing we have to do is stabilize basal transcription apparatus binding. And for that, we invoke another DNA binding spot, something that is called an enhancer. So this is a strictly eukaryotic idea. And the enhancer is a DNA sequence, and it binds a generic term we call a transcription factor. Let's call this, though, today, a transcriptional activator. There are some repressors, but most of the time it's activators. A transcriptional activator. And what that's going to do is allow for a more efficient binding of the basal apparatus and a more efficient binding of RNA polymerase. So, enhancers have a very specific role and very specific definition of way we can identify if a DNA sequence is acting as an enhancer or a promoter. And it just occurred to me, a really good question would be, how can you distinguish between an enhancer and a promoter? And we do it in the following way. A promoter is essentially the binding site for RNA polymerase to establish where +1 is going to be and what direction you're going to go. So a promoter, this is distance and orientation dependent. That means that you set the promoter system down, it establishes what +1 is going to be, and it says you're going to start transcribing that way. So really, by doing recombinant DNA work, you can turn any chunk of DNA into a gene. You can make any chunk of DNA get expressed. All you got to do is put a promoter system right wherever you want it, and say, okay, this is where we're going to start expressing RNA from this spot. And you're also directing the specific direction you're going to go. We've been looking at, with our little sequencing analysis, we can have genes that run in two different directions. So, that's the promoter. On the other hand, we have the enhancer, and the enhancer's job now, it's binding a transcription factor, but the way it's going to work is going to cause a folding over of the DNA to allow for a more efficient binding of the basal apparatus. And we can then say that enhancers are distance and orientation independent, because they can fold around from different sides of the gene. They are not directing which way, or essentially which strand the RNA polymerase is binding to. They simply are increasing the efficiency by which RNA polymerase can bind to its promoter. So it turns out then in eukaryotic genes, you have to have the promoter, of course, and then you're going to have the enhancer. And enhancers offer the ability to have very specific transcription control, in that the transcription factors, the transcriptional activators, this affords us a system by which we can have a tissue-specific transcriptional activator. We have, for a whole range of hormone reasons that we'll be getting into in cell signaling discussions, a transcription factor that's present in one cell that's not in the other, and that means that there's going to be gene sets that can be activated in this cell that are not going to be active in another cell. So it allows us for differential gene expression this way. Yeah, there you go. So the number of questions on this is probably going to be fairly light, but some kind of good sort of dissecting a promoter in a bacterial system, something like that, and maybe defining promoters and enhancers and distinguishing between the two. Thank you. All right. The bigger part of what we've been talking about was some membrane mechanics, and one of the primary reasons by getting into membrane mechanics was to be able to establish the notion of an impermeable membrane, and establish the idea that by having a let's see, what's the term I would use? Anyway, a disequilibrium of stuff, glucose, protons, sodium, on one side versus the other of that membrane, we actually create organization, we create energy that way, and we can use that to drive different kinds of reactions, and we can establish these different kinds of concentrations within a cell and its environment. So we can sort of establish this chart of looking at membrane transport. I guess the first thing we should establish is that a good phospholipid bilayer, one that we haven't mucked with drugs, membranes are, for all intents and purposes, impermeable to ions. Any kind of charged molecule ain't getting through. A charged molecule that ain't an ion, like glucose, these things are impermeable. And then we've got things that the membrane is permeable to. And this is going to be things like O2 and CO2, to a degree water, sort of organic hydrophobic molecules. These things can transfer across the plasma membrane. And, that's an important distinction then. So there's a lot of stuff that we need to bring into or push out of the cell that is going to require some kind of facilitated system, some sort of protein system. And then we got stuff that just diffuses. And so that brings us to like the next level of definition. And we can define diffusion simply as a thermodynamic process, the movement of molecules from an area of higher concentration to an area of lower concentration. So, diffusion is absolutely just a thermodynamic principle. Now, one of the things that plays into this is by monkeying around with diffusion, we can actually then bring in this concept, the idea of osmosis. And this is still simple diffusion, but it is the diffusion specifically of water across a membrane that's not permeable to everything, particularly ions. So we call the semi-permeable membrane. All right. So this allows for the movement of water, but only under certain conditions, i.e., water is following its diffusion gradient. And so, we can think of it in a couple of different ways. First, water is going from an area of higher concentration to an area of lower concentration. That's easy. But another way to keep that in mind is simply this. Water follows salt or sugar, various dissolved osmotica. But water will move towards salt. So you don't pump water, you pump salt. Water will follow. All right. So once we start thinking about moving molecules that have a charge, things that can't move through simple diffusion, that's where our chart now comes into play. And we've got then our membrane transport chart. And we can, on one side, look at passive transport and define passive transport. Can you see that? Not so well. Maybe I'll use a different marker. Blue seems like it's got pretty good contrast. Let's try the blue. All right, so we have passive. And this is going to be using a diffusion gradient. So passive transport, we're going from a high concentration to low concentration. We have a couple of different kinds of passive transport systems. We have just plain old diffusion, would be put over on this side. But then we also have what is known as facilitated diffusion. And under this, we got a couple of different subheadings. We've got things like a channel that is not particularly picky about exactly what's going through it. It's based on size and charge, but it's not overly specific. And the channel can be a gated channel. And we'll fill out the different kinds of gates when we get to the nervous system in a moment. And then we also have, with the passive transport system, a passive transporter. And the transporter now is going to be acting a little bit more like an enzyme, in that it is significantly more saturable, and it is going to be quite specific. An example of this is the glucose transporter, the glucose passive uptake system. And it is binding glucose and allowing it to go into the cell, it only binds glucose. It won't even bind the stereoisomer. And if you end up, it's got a maximum rate of stuff they can transport. And so there comes a point where it can't move things any faster. And so the uptake of glucose by the glucose passive transporter resembles an enzyme catalyzed reaction kind of rate. And then if we look at here the rate of glucose uptake, we see something like this. And so this is the glucose concentration, in which we got a maximum velocity, and we can actually go through the entire KM process, and people do. All right, this is because the number of glucose transporters is fixed on the cell surface at any one moment. Actually, insulin can induce the number of glucose transporters, and so you can cause glucose to be taken up at a higher or lower level. But with a given set of glucose transporters, you got a fixed amount of glucose you can bring into the cell. So this is the transporter difference. Now, on the other side, we've got active transport, and we break this down into two different forms as well. We have direct active, I guess I should say here, active is going from a low concentration to a high concentration. That is, it's accumulating stuff against its gradient, and so you're violating the first or the second law of thermodynamics here. Direct is going to use ATP hydrolysis. Adding that phosphate, that big negative charge, changes the shape of the protein, and that's what's going to drive this system's ability to bind something with a high affinity, open up on the other side of the membrane, change binding affinity, and it falls out. So we have direct, which uses ATP hydrolysis, and then we have an indirect active transport. It's still accumulating stuff, but instead of using ATP directly, we're now going to be using some kind of existing gradient to drive uptake of the second molecule. So we've got a couple of different examples of this. Of course, the number one direct active transport system is the sodium potassium ATPase. And the thing to always keep in mind, and I'm not sure what mnemonic you might use, other than the fact that sodium is always high outside. I'm sure we could have some kind of strange references to some dude named Sodium sitting outside. Regardless, we're always pumping sodium out, potassium in. Sodium is always real high concentration by 2 or 3 orders of magnitude higher outside than it is inside. And we can use then that existing sodium gradient to drive indirect transport. And so we have the sodium potassium ATPase. On this side, we have the sodium coupled glucose transporter. And so the question then always can be, if you block the sodium potassium ATPase, you're also going to block the sodium coupled glucose transporter because the gradient's not there. We throw in the glucose transporter, but pretty much everything that you're going to bring into your cells, all the amino acids, all the various vitamins that are not fat soluble, all kinds of things that you're bringing into your cell, you pretty much are bringing in with this sodium cotransport system. So it's like a generic system that you can always depend on for energy. Is you always know sodium's going to be higher on the outside than the inside, so if you need to bring something in against its gradient, just use a sodium coupled cotransport system and we got you covered. So a couple of things fall from this. If we block the sodium potassium ATPase, if we inhibit that, and there's some drugs that'll do that, we inhibit the cell's ability to pump salt out. When you do that, there's still co-transport going on up until you no longer have a sodium gradient. So sodium is going back into the cell, and we actually end up making a cell that itself is hypertonic to its existing environment, has too much salt, and water starts moving in. So blocking your sodium-potassium ATPase is a quick way to screw yourself up. And, you know, it is so important that right now you're using 30% of your metabolic energy to do nothing but pump sodium out. All right, so that's good. From there, we'll just kind of back up and pick up on some gated transport systems, looking at the signaling and nervous system signal production in a neuron. And here we invoke the idea of the different gates. We have the voltage-gated neuron, or excuse me, the voltage-gated ion channel, the ligand-gated ion channel, and then the old mechanosensory-gated ion channel. And combining these guys, we can make a neuron operate. So, just as a little refresher, we've got a neuron. And so, pretty much, this is everything you need to know about neuroanatomy in one little diagram here. We've got the cell body. Yeah, you can see that. The cell body contains all these projections coming out. It also has the nucleus. We have this long, very long projection. This is the axon. These are dendrites. And we send a signal using a membrane depolarization event. So rather than changing the charge of the whole thing, which we really couldn't do fast enough, we have little sections of this thing that are depolarizing. And we'll just go ahead for now say we triggered a depolarization event. And what we have happening then with membrane depolarization we've got a change in voltage, which is essentially a change in sodium ion concentration in patches of the cytoplasm. As the voltage changes, it triggers the next patch. And so this is where we're using the voltage-gated ion channels. They are essentially sodium ion channels. And there's also potassium leak channels as well, but the big guy is sodium ion channel, which responds to a voltage change. And so we go from, let's see, yeah, we'll do it this way. So minus 60 millivolts down here. Actually, we'll put this line up here at 0, not that anybody cares. But I'm on camera, you know? And so we go at minus 60 volts here. We have the voltage change that causes the voltage-gated ion channel to snap open. Sodium rushes in, the voltage goes up to plus 40, and then as the potassium leaks out, it goes back down, undershoots, and comes back up. And so this is potassium leaking out. But at the end of this little pulse, your ion concentration is completely screwed up. You got a ton of sodium inside, a bunch of calcium or potassium outside. And so you now have to take a few moments to sodium potassium ATPase your way back to normalcy. And so right here, you can hear, if you listen very carefully, the sodium potassium ATPases are cranking up like crazy. They roar up to life. They very quickly resettle the ion balance, and we're back again ready to take another neuronal impulse. So this allows the voltage change to travel down the membrane, because the patch here is completely discombobulated. The sodium and potassium gradients are completely screwed up at this point. And so the voltage gradient, or the voltage change, is going to trigger this next patch. And so we just go sort of chunk at a time all the way down to the end. We get down to the end, of course, this is where we have the little fluid gap or air gap. That is the synapse. So okay, what we'll do is erase here. And diagram at high mag. There's the end of the axon. And here is the connecting cell. Might be a muscle cell. We have the neuromuscular junction. Might be a, you can't see that all that well. We'll go back to my dark blue. It might be another neuron in your brain. We got neuron to neuron connection here. And this gap is the synapse, the synaptic cleft. And we're going to have vesicles filled with little neurotransmitters. And when the voltage change hits here, it causes these synaptic vesicles to pop loose from their anchors. They fuse with the membrane, and the neurotransmitter comes out. So on the postsynaptic neuron, or postsynaptic cell, muscle cell, whatever, we're going to have here our ligand-gated ion channels. And as we finished up, when we were talking about this, in the simplest way, it's going to be another sodium ion channel. That opens up, starts the whole depolarization thing again. That would be an excitatory signal. You also can have, this gets into the complexity of neurobiology, you can have a chloride ion channel that opens up, dumps chloride in, and now you make this thing even more negative, and that would be an inhibitory signal. But that's the complexity of neurobiology. We won't get into that. So that's how we can begin using ligand-gated ion channels. Mechanosensories are essentially things that allow you to have pressure, photoreceptors, hearing. And I was actually, while I was doing this, I was thinking about a Venus flytrap, which, if you've never seen one, they actually are quite cool. And maybe you have seen like touch-sensitive leaves, where you touch them and they go whoop, and they zip in. And what you're doing here is you're actually operating, or the little bug is operating and pushing on a mechanosensory gated ion channel. And it ain't sensory, it's mechanic. It is just a pushy-pull type touch-sensitive ion channel. That ion channel opens up, and in plants it's probably potassium, but regardless, you're changing the ion concentration, and now we have ions moving from one cell to another. And as we say, you don't pump water, you pump salt. And so by shifting the ions from one cell to another, we shift water flow, and the cells that are at the hinge point of that leaf are wrapped in cellulose in such a way that as the ones that lose sodium begin to also lose water, and the ones that gain sodium begin to gain water, when they start to swell up or shrink, they're going to actually start bending and kind of going from like being a lozenge shape or a burrito shape to curving up. And you have enough of these cells arranged in just the right way. And so, in Batman and in general sort of concern, I was always worried that, trekking in the jungle, you'd stumble into a giant Venus flytrap and it would blam, clamp on top of you. It was the same level of concern I had for quicksand. But that movement is significantly slower, because you do have to now have the mechanosensory or the mechanical gated ion channel open up Sodium's moving around, and then it's going to take a while for the water to move in, expand that cell, and start actually causing it to close. So it's not going to be as fast as a neuronal transmission. It takes a little while, but it does start to eventually move on you. And so I figured then you have time to, you step into it, but it's not going to grab around your leg and, engulf you. That's pretty much it. Let's see. Yeah, I like that. We went through, we kind of did this a little bit backwards because we've been going through all the metabolism stuff. But there you go. So in terms of studying for this, what you want to do is sort of organize your thoughts into these different kinds of tables that we've been doing. You got your transport table. And by filling in the details and kind of learning it from that direction, you're giving yourself the basic outline. And that'll give you the ammunition to answer a bunch of questions. There are going to be some questions that you may have to think about, okay, what's going on here? Why would this be happening based on what we know? But you'll be able to get a fair amount by just having these charts sort of set in your head, i.e., compare active versus passive transport. How do you define them? Why is the sodium-potassium ATPase critical for glucose uptake, the indirect active transport of glucose? Or another way of saying, okay, talk about indirect glucose active transport using the sodium cotransport system. And then sort of a question that is a follow-up. What's the role of the sodium-potassium ATPase in this? And the answer to that is the sodium-potassium ATPase is always making sure that the cytoplasm, the inside of the cell, has got low sodium concentration. So no matter what you're doing, you could be using a facilitated transporter to be pushing glucose with its gradient, and then you could be using a indirect active to be pulling it in. As long as you maintain the appropriate sodium concentration, everything's going to be working just fine. I mean, this is like the classic example is in a gut cell, in that the lumen of your intestines has a fair amount of sodium compared to the inside of the cell. So we've got the sodium cotransport system that's bringing it in. Then the cell has got sodium coming in, so the sodium transport system has to pump it back out to always maintain that gradient. And it's sort of like a generic tool that you can depend on sodium concentration being low. And so you have a third-party contractor building this transport system, and it's like, yeah, I don't know what the conditions are going to be, but I can guarantee you that sodium will always be low. So if you rig your system such that it's using a sodium diffusion gradient, we can guarantee you that the inside of our cytoplasm will be low. And, that's how you build it. All right. Let's see, what else would we ask? Then, the level in terms of metabolism, breakdown of stuff, what goes in, what comes out, a few questions based on that. Some definitions, probably no real structural diagrammy things. That's all I know. Good. Always to say, if you're still watching, what, you expect previews? Yep, let's plan on the exam dropping Monday morning, 8 a.m. And if you want to, print it out and hand it to me, I love that. I'm going to have to print them out anyway. So if you're around in the building or whatever, give it to me. All right, that's all I know. Actually, it's fine. Post it to Moodle, whatever. Bye-bye.